Welcome back to the Deep Dive. Ready to explore some more fascinating science? Today it's all about pharmacodynamics.
Pharmacodynamics.
But we try to keep things kind of understandable here, right? We
I
don't
like
want to
it.
bore everyone with a medical school lecture, so
No textbooks here.
no textbooks, exactly.
Just the cool
Yeah,
stuff.
just the coolest stuff about what your medicine is actually doing once it gets inside you. We've got some great material to pull from for this deep dive. And like I said, we're going to try to highlight the good stuff.
Yeah, the most important stuff.
Yeah, and keep it interesting.
Exactly.
So let's get right into it. Where do these drug molecules even go? Like, what are they targeting?
Right to the point.
Yeah.
So they're actually targeting specific molecules in your body, and one of the biggest categories of those is receptors.
Receptors.
Yeah, and you can think of receptors like proteins that are on your cells or sometimes inside your cells.
Okay.
And these receptors are kind of like locks, and the drugs are like keys.
Ah, okay.
So the drug will bind to that receptor,
Gotcha.
and that binding triggers a signal, kind of like a domino effect.
Oh, I see.
Now, what's interesting here
Yeah.
is that this interaction, it's usually very specific, meaning the drug will only fit a very particular receptor shape.
You can't just go to any receptor?
No, not just any receptor. It has to be the right one.
Oh.
Another important thing is that it's what we call saturable,
Okay.
which means there's a limited number of these receptors available.
It's like musical chairs.
You got it. Like musical chairs, the drug can't bind if there's no receptor available.
I see.
And the last thing, it's reversible. So the drug doesn't usually stay bound to that receptor forever.
It comes on and off.
It comes on and off. Exactly.
So that all matters for dosage.
Oh, absolutely. Dosage is super important. That's why
Right.
that's why doctors spend so much time figuring out exactly the right amount of medication.
To give you...
Yeah.
Okay, so we got these receptors. What other targets are there?
Well, we've got enzymes.
Enzymes, right?
Yeah, you've probably heard of those. Enzymes are these proteins that speed up chemical reactions
Okay.
in your body.
Mm-hmm.
And some drugs actually block these enzymes.
But lock them.
Yeah, they stop them from working.
Okay.
Like, you've probably heard of NSAIDs.
Oh, yeah, like ibuprofen.
Exactly, like ibuprofen or aspirin. Those are NSAIDs, and they block an enzyme called KOX,
Both.
and that helps reduce inflammation and pain.
Okay.
Now, sometimes drugs can actually increase the activity of enzymes, but blocking is more common.
Oh, interesting.
Yeah, it is. Then we've got ion channels.
Ion channels.
Yeah.
Okay.
These are kind of like little gates that control the flow of charged particles.
So they're controlling the flow of these particles.
Yeah, in and out of the cells. And this affects electrical signals, especially in nerves.
Interesting.
A good example here is local anesthetics.
Oh, okay.
Like when you go to the dentist,
Right.
they numb you up. Right?
Yeah, that's right.
That's because those drugs are blocking sodium channels, which basically stops the pain signals from traveling up the nerves.
Wow.
Yeah, pretty cool.
Okay, so those are kind of the big ones, right? Receptors, enzymes, ion channels.
Those are the big three.
Are there others?
There are a couple more, but they're not as common.
Okay.
Like we've got transporters, which move substances across cell membranes. A good example here is SSRIs. Those are antidepressants that work by blocking serotonin reuptake.
Blocking it.
Yeah.
So the serotonin stays where it is.
Yeah, basically it increases the levels of serotonin in the brain because it's not being transported back out.
Okay, cool.
And then there are some less common targets like structural proteins,
Structural proteins.
like tubulin, which is important for cell division. Some anti-cancer drugs target that.
Oh, wow.
And even less common are drugs that directly interact with DNA or RNA.
Directly with the DNA.
Yeah.
Wow.
Those are usually antiviral or anti-cancer drugs that are trying to mess with the genetic material of the virus or the cancer cells.
I see. So now that we know where the drug goes, how does it actually do anything?
Okay, so once the drug reaches its target, it's all about these chemical forces between the drug and the target. We talk about affinity, which is basically how strongly the drug binds to that target.
Okay.
And we measure that with something called the dissociation constant, or KD.
KD.
Yeah. Now, a lower KD means a higher affinity, meaning it's a tighter grip.
So the lower the number, the better.
Yeah, you could think of it that way. But just binding isn't enough. The drug also needs what we call intrinsic activity.
Intrinsic activity.
And that's the ability to actually activate the target and produce a biological response once it's bound.
I see. So it's got to do more than just stick there.
Exactly. It's got to do something.
Okay.
Now, there are some drugs that just block. They don't activate.
Okay.
Those are called antagonists.
Antagonists.
But there are drugs that both bind and activate, and those are called agonists.
Okay. Agonists, antagonists.
It's exact.
So agonists are like the ones that actually make things happen.
They're like the body's natural messengers. They
Uh-huh.
bind and they activate.
I see.
Now, with antagonists, they bind to the receptor,
Good.
but they don't activate it.
So they're just kind of in the way.
They're in the way. They're blocking the natural messengers or other drugs from binding and having their effect.
Oh, okay.
And there are different types of antagonists. There's competitive antagonists,
Okay.
which bind to the same site on the receptor as the agonist.
They're fighting for the same spot?
They're fighting, exactly.
Okay.
And then there are non-competitive antagonists,
Okay.
which bind to a different site and kind of change the shape of the receptor so the agonist can't bind as well.
Oh, sneaky.
Very sneaky.
All right, so we've got those two. Are there any other types of drugs that interact with these targets?
Yeah, so there are some in between.
In between.
Yeah, like partial agonists,
Oh.
they bind and they activate.
Okay.
But even at the highest doses, they can't produce the full effect like a full agonist can.
So they're like a weaker version.
You could think of it that way, yeah.
Interesting.
Then we've got inverse agonists.
Inverse.
Yeah, these act on receptors that have some kind of baseline activity,
Okay.
even without anything bound to them.
Oh, so they're always a little bit on.
Yeah, they're always a little bit on, and these inverse agonists can actually reduce that activity.
Oh, wow. So they turn it down even further.
They turn it down.
Gotcha.
Then we have allosteric modulators. Those bind to a different site than the main drug binding site, and they can either boost or reduce the binding or the effect of the main drug.
So they're like the helpers.
The helpers or the hinderers, yeah.
Okay, so how do doctors figure out the right dose? Like how much medicine to give?
So we look at what's called dose-response relationships, and we often visualize this with something called a dose-response curve, which is usually S-shaped. And from that, we can measure things like potency,
Okay.
which is represented by the EC50 or ED50.
EC50.
And that's the concentration or the dose of a drug that's needed to produce 50% of its maximum effect.
Okay.
And the lower the EC50, the more potent the drug is, so you need less of it to get the same effect.
Interesting.
Then there's efficacy.
Efficacy.
Yeah, or
Dose.
Emax, which is the maximum effect a drug can achieve no matter how high the dose.
Oh, I see.
So some drugs might be really potent,
Okay.
but they might not be able to produce a very strong effect.
So you need a little bit to get some effect, but you can only get so much effect.
Exactly.
All right. And of course, safety. We want to make sure the drugs we're taking are safe.
Oh, absolutely.
How do we measure that?
So there's the therapeutic index, which we call TI.
TI.
And that's the ratio of the dose that produces a toxic effect to the dose that produces a therapeutic effect.
Okay.
And a larger TI generally means a safer drug.
So bigger is better?
Bigger is better in that case, yeah. And then there's the therapeutic window,
Okay.
which is that range of doses where you get the desired effect without too many side effects.
Oh, that sweet spot.
That sweet spot, you got it.
Okay, so the drug binds to its target.
Uh-huh.
And then what happens inside the cell?
Well, that's where signal transduction comes in.
Signal transduction.
Yeah, and this is basically a cascade of events
Okay.
inside the cell,
Okay.
triggered by that initial interaction between the drug and the target.
I see.
And it often involves these signaling molecules called second messengers.
Second messenger.
Yeah, like CAMP or calcium. And these messengers trigger all sorts of changes in cell function.
Wow, it's like a whole chain reaction.
It is. It's like a chain reaction.
Yeah.
And some drugs can even affect gene regulation by binding to nuclear receptors, which then move into the cell's nucleus.
To the nucleus.
Yeah.
Okay.
And those receptors can actually turn genes on or off.
Wow. So they're controlling what the DNA is doing.
They're influencing it, yeah.
That's amazing. So what happens if you're taking more than one medication? Can they interact with each other at this level?
Oh, absolutely. We see that all the time. We call them pharmacodynamic drug interaction.
Drug interactions.
And sometimes we see additive effects, which is where the combined effect of the two drugs is just the sum of their individual effects.
Okay.
1 + 1 = 2, basically.
Gotcha.
But sometimes we see synergistic effects, where the combined effect is greater than the sum of the individual effects.
So 1 + 1 = 3.
Exactly. You get a boosted response.
Wow.
And then there are antagonistic effects where one drug can actually reduce the effectiveness of the other drug.
They're working against each other.
They're working against each other. That's right.
Wow. So that's why it's important to tell your doctor about everything you're taking.
Oh, absolutely. It's crucial.
Okay, last question. Why does the same drug work differently for different people?
That's a great question, and it's all about individual variability.
Individual variability.
And a lot of factors contribute to that, like genetics, for example.
Like our DNA.
Our DNA, yeah.
Yeah.
That plays a huge role in how our bodies handle medication.
Well,
And then there's age. Older people often process drugs differently than younger people.
makes sense.
And then any existing diseases can also affect how a drug works. The body's in a different state.
Right, right.
And of course, other medications you're taking,
Like we just talked about.
like we just talked about, those interactions are really important to consider. And even things like diet and lifestyle can play a role.
Wow, so many factors.
It's complex, but it's super important to understand.
Absolutely. This has been a fascinating look at pharmacodynamics.
It's a fascinating field.
It's amazing to think about all these tiny interactions the molecular level.
It really is.
And, all the things that can affect how our medications work.
So many things to consider.
It really makes you think about the future of medicine. With all this complexity and all this variability between people.
Yeah.
Like, how can we design and use drugs even more precisely
That's the big question.
to get the best effect with the least side effects for everyone?
Absolutely. That's the goal.
Well, on that note, thanks for joining us for another deep dive.
Always a pleasure.
I'll see you next time.
See you then.